Let's talk about central dogma. What is central dogma? Central dogma is the process by which the instructions in DNA are converted into a functional product. It was first proposed in 1958 by Francis Crick, the discoverer of the structure of DNA. Central dogma of molecular biology explain the flow of genetic information from DNA to RNA to make a functional product, protein. The central dogma suggests that the DNA contains the information needed to make all of our proteins, and the RNA is the messenger that carries this information to the ribosomes. The ribosomes serve as a factories in the cell where the information is translated from a code into a functional product. The process by which the DNA instructions are converted into the functional product is called gene expression. Gene expression has two key stages, the transcription and translation. In transcription, the information in the DNA of every cell is converted into a small portable RNA message. During the translation, this message travels from where the DNA is in the cell nucleus to the ribosomes where they are read to make specific proteins. The central dogma states the pattern of information that occurs most frequently in our cell is from existing DNA to make new DNA is DNA replication. From DNA to make new RNA is transcription, and from RNA to make proteins is translation. First, we discuss the DNA replication. Before we start the DNA replication, what is DNA replication? It is the biological process in which cells create two identical copies of DNA from the original DNA molecule with a high accuracy fidelity rate. DNA replication is a semi-conservative process, which means each strand of DNA molecule acts as the template for the synthesis of a new complementary strand. This process takes one starting DNA molecule to two daughter molecules with each new DNA double helix containing one new and one old parent strand. The replication of DNA begins at the sequence of nucleotides called the origin of replication. Helicase unwind the double-stranded DNA helix, and the single-strand binding protein reacts with the single-stranded regions of DNA and stabilize. DNA polymerase III is the major enzyme involved in DNA replication. DNA polymerase III can only add nucleotide to the 3' end of pre-existing chain of nucleotide and cannot initiate a nucleotide chain. Therefore, a RNA polymerase called primase constructs RNA primer, a sequence of about 10 nucleotides complementary to the parent DNA. DNA polymerase III can add deoxyribonucleotides to synthesize new complementary strand of a DNA. Because the two parent strands of DNA are antiparallel, they are oriented in opposite direction and must therefore elongated by different mechanisms. The leading strand elongates towards the replication fork by adding nucleotides continuously to its growing 3' end. In contrast, the lagging strand, which elongates away from the replication fork, is synthesized discontinuously as a series short segments called Okazaki fragments. When the DNA polymerase III reaches the RNA primer on the lagging strand, it is replaced by the DNA polymerase I, which removes the RNA and replaces it with the DNA. DNA ligase then attach and form a phosphodiester band in the DNA's further unwind. The new primer are made and DNA polymerase III jumps ahead to begin synthesize another Okazaki fragment. For simplicity, DNA polymerase III has been picked separately units, one acting the leading strand and the other acting in the lagging strand. The current view of DNA polymerase III is that two subunits function together with the DNA on the lagging strand folding to allow the dimeric DNA polymerase molecule to replicate both strands of parental DNA duplex. DNA proteins other DNA polymerase III are not shown. So this is the last part of DNA replication.
Let us talk about transcription and translation. Keep in mind that transcription comes first before translation. Transcription has letter C in its word and translation has L on it. And in alphabet letters, C always comes first before letter L. Here, we will understand DNA or how these molecules code for a particular organisms. How is that a single cell containing a specific set of genetic material will result to a development of a fish, cat, or human? To understand this phenomenon, we'll need to learn about transcription and translation. Transcription and translation. This is a collective process by which genetic code is read by enzymes in order to produce all of the proteins in an organism. A very long chromosome contains a certain base pairs, which is called as genes. This is the whole. This whole is the chromosome, and these base pairs are the genes. Genes are responsible for coding of different things. When gene is expressed, a specific protein is produced. The first step is called transcription. As I've said, transcription is always comes first before translation. Transcription is a process by which enzymes uses one of the DNA within a gene as the template to produce mRNA or messenger RNA. To do this, RNA polymerase with the help of protein called transcription factors binds with a specific sequence within a gene called the promoter and split up the two strands apart. One of the strand is the template strand or the antisense strand. This is the template strand or the antisense strand. And the other part is the non-template strand or the sense strand. And this one at the lower part is the non-template strand or the sense strand. RNA polymerase doesn't need a primer. It simply initiates RNA synthesis at start point and then move downstream along the gene in a process called elongation, synthesizing the mRNA as it goes. It will move downstream here. From here, and it moves downstream. Reading the antisense strand from 3' to 5' end, generating the mRNA to 5' end, attach nucleotide to 3' end as it goes. RNA polymerase zip the DNA back up as it goes. It will zip the DNA strand as it goes. Here. Once the RNA polymerase reaches the end of the gene termination occur. It says that once the RNA polymerase, this RNA polymerase, when it reaches the end of the gene, termination will occur. The enzyme detaches from the gene and the DNA return to its original state. It will this RNA polymerase will detach, and this mRNA produced will also detach from the template strand. And after a quick modification, this mRNA leaves the nucleus and move to the cytoplasm and find the ribosome. This is where the translation occurs. Here. This mRNA will leave the nucleus. This is the nucleus, as you see. This mRNA will leave the nucleus and move here to the cytoplasm and find the ribosome. The ribosome, where the translation occurs. During translation, the mRNA acts as the protein code, which has 3 sets. Each code has 3 sets in its base, which called the codon, that will code for the specific anticodon carried by specific tRNA or transfer RNA. Each tRNA is attached to a specific amino acid. These 3 letters are the codon. Next. And this codon at the lower part are the tRNA. mRNA is the codon and the tRNA are the anticodon. Next. AUG are usually the start codon, and it codes for an amino acid which is called methionine. And UAA, UAG, and UGA are the stop codon or the termination codon, as it do not codes for any amino acid. The translation occurs at the ribosome. The small subunit bind the mRNA and an initiator tRNA which adhere to start the codon. Then the large subunit join the translation initiation complex. Then the tRNA is and the tRNA that correspond to the next codon after the start codon will enter the ribosome. This will carry with it an amino acid which become covalent bond to the methionine from the initiator tRNA. The first tRNA detaches and leaves the ribosome, which has shifted over, making room for the next tRNA. And this new amino acid link to the first two, and the process continue all the way down the mRNA. It will continue all the way down. As the tRNA comes, enter and exit the ribosome in a sequence that is dictated of the codons on the mRNA, a polypeptide chain will grow. This continue until the stop codon is reaches, or the stop codon has been reached. This anticodon UAG, stop codon or the termination codon has been reached. The process will end. At this point, the completed polypeptide will float away for folding and modification. Here, the finished polypeptide will float away for folding and modification. Float away from the ribosome. For clarification, transcription is to transcribe DNA to mRNA. And mRNA is translated into protein. Transcription process which process DNA to mRNA, and translation processes mRNA to protein. All simply by obeying the base pairing occur in a nucleic acid, gene code for specific protein, and protein makes of most of what organism composed of. From muscle tissue, organ tissue. This is how DNA carries a code for an organism. Thank you.